Well, hello, my fellow pre-meds. In this video, we're going to mainly focus on the structural hierarchy of proteins, although we will also touch on the peptide bond and denaturation of proteins. And although the concepts discussed in this slide deck are not particularly complex, they're not very difficult to digest, they do show up quite frequently on the MCAT, and therefore I thought it was necessary to include this video as part of this biochem playlist. And if you use our associated Anki cards, you'll find that these points will be easy points on the MCAT. These videos, at least as of now, are sponsored by you. So if you're interested in supporting me to be able to continue to make these high quality MCAT playlists, you can support me either via Patreon or by buying me a coffee. Links in the description down below, as well as the links for everything else, including the link to our growing pre-med companion Discord community. Let's jump right in. When we think about protein structural hierarchy, we think about four broad categories: the primary, secondary, tertiary, and finally the quaternary structure. So we're going to get into each of these on subsequent slides. The primary structure of proteins is the sequence of amino acids that comprise said protein. So these amino acids are joined together via peptide bonds. So if you look at our little cartoonish schematic over here, this is a polypeptide sequence, or the primary structure of a protein. And you can see the individual amino acids joined with one another via these peptide bonds. Now, you can determine the sequence, you can analyze the sequence of these polypeptides using lab techniques such as cation exchange chromatography and Edmonds reagent, as we'll see in upcoming slides. And then finally, an abnormal amino acid abnormal amino acid sequences are implicated in many genetic diseases. So one that comes to mind is sickle cell disease. And indeed, sickle cell disease does show up quite frequently on the MCAT. So here's what happens in sickle cell disease. You have an amino acid residue that's supposed to be polar is exchanged for one that is hydrophobic. So if you recall from our amino acid slide deck, glutamic acid has a polar charged side chain, whereas valine has a hydrophobic side chain. And essentially what happens is you lose a glutamic acid residue and it's replaced by valine, and that throws off the entire structure of hemoglobin, and therefore it loses its function, its normal biological function, and that kind of causes the disease state. All right, before we move on to the secondary structure of proteins, we're going to talk about the peptide bond. So the peptide bond is formed by the amino group and carboxyl groups of two amino acids. The peptide bond is planar. You can see it right here. So this is serine, this is alanine. You can see the peptide bond joining them together. And then the peptide bond actually has partial double bond characteristics because of resonance over here. So therefore, it won't be able to rotate about its axis. Notably, the peptide bond, while uncharged, is polar. So recall from previous videos that the amino group and the carboxyl group of amino acids are capable of carrying charges. Well, once those amino groups and carboxyl groups join to form a peptide bond, they can no longer accept or donate protons. However, they are still polar, as you can see right here. This carbonyl group has partial negative charge on the oxygen, and the amino group has a hydrogen atom that's capable of participating in hydrogen bonding. So while they won't carry any charges, they can certainly participate in hydrogen bonding, and this will become very important when we talk about the secondary structure of proteins. Next, we have the fact that these peptide bonds are typically in the trans configuration, meaning that the side chains of the amino acids will be on opposite sides of this peptide bond. And that's because you don't want to have two bulky groups on the same side. And then finally, by convention, the peptide bond is sort of read from the N to C terminal. So the N terminal is the amino terminal, and the C terminal is the carboxyl terminal. All right, now let's talk about the secondary structure of proteins. We have sort of three flavors over here. We have the alpha helix, the beta sheets, and the beta turns, or the beta bends. All right, so the alpha helix is formed from a single strand of a polypeptide chain. So we'll distinguish that with the beta sheet that has multiple strands typically. And then the way it's linked, the way you form a secondary structure is via hydrogen bonding. So hydrogen bonds will link the backbone of every fourth amino acid. What does this mean? So like, let's say this is one amino acid right here. You count 1, 2, 3. So this is the fourth one. So this fourth amino acid is going to interact with our first amino acid via a hydrogen bond, and so on for the rest of the alpha helix. So over here it says I to I plus 4, that's basically amino acid to amino acid plus 4, or the fourth amino acid. And by the way, this hydrogen bonding occurs between the carboxyl groups and the amino groups in the backbones of amino acids, and notably not in the side chains. So even if you have a polar side chain, it's not the one contributing to the hydrogen bonds in the case of a secondary structure or in the case of an alpha helix, because as you can see, the R groups are pointing outwards. Okay, next we have that proline can introduce kinks in alpha helices, and essentially the idea over there, if you watched our slide deck on amino acids, proline contains its backbone amino group embedded within a ring, and because it's embedded within a ring, it won't be able to participate to a great degree in this hydrogen bonding, and therefore it'll introduce kinks in the alpha helix. That's very high yield on the MCAT. All right, now let's talk about beta sheets. So beta sheets are composed of 2 or more strands. So don't get this confused. They're not composed of 2 separate polypeptide chains. Rather, it's the same polypeptide that's forming multiple strands, okay? Because if it were 2 separate polypeptide chains, then it would be a quaternary protein, as we'll see soon. Now, these sheets will have a pleated appearance. I didn't really know what a pleated appearance is, but apparently, like how some skirts will have this appearance, that is the pleated appearance that beta sheets show. And again, this is stabilized by hydrogen bonds that occur between 2 separate strands. So as you can see right here, this is one strand and this is another strand. The hydrogen bonding is occurring between 2 separate strands, so we call this interstrand bonding, whereas in the alpha helix, because we only had one strand, we call it intrastrand bonding. And then sometimes you'll see beta sheets represented as arrows, as you can see over here. And these turns over here, these loops, are what we call beta bends or beta turns. And this is where you will frequently encounter proline, because recall proline caused the kinks in the alpha helices. Well, this is where you can typically find proline. All right, next let's talk about the tertiary structure of proteins. So the tertiary structure for many proteins is the final 3D conformation that you will have that will carry out its functions. Although some proteins have a quaternary structure, as we'll see in a bit. So what determines how a protein is going to fold on itself? Well, it has nothing to do with the backbones of amino acids. So recall thus far in the primary and secondary structures, what joined them together and what formed them were the backbones, the amino and carboxyl groups in the backbones of amino acids. Well, in the case of the tertiary structure, what's going to determine how the protein folds on itself is the primary sequence of amino acids, specifically the side chains of that primary sequence. So recall how we looked over here at the polypeptide chain. Well, whatever side chains are going to be over here are going to determine what the final 3D structure will look like. And that's because, if you have a bunch of hydrophobic side chains, then you will get hydrophobic interactions that will determine the final 3D structure. If you get a bunch of charged amino acids such as glutamic acid and lysine, for example, you'll get ionic interactions that will determine the final structure. Or, for example, if you have a bunch of cysteine residues, as we discussed in our amino acid slide deck, they will form disulfide bonds, and that will determine the 3D structure. Now, in reality, it's a combination of all of those. It just depends on which ones you have more of and which ones you have less of. Notably, the 3D structure in the tertiary structure of a protein is composed of one subunit or one polypeptide. If there's more than one polypeptide, then that is going to be a quaternary structure. So if you have multiple polypeptides or multiple subunits joining with one another, then you have what's called a quaternary structure. And again, they're joined together by similar interactions as the tertiary structure, so disulfide bonds, hydrophobic interactions, hydrogen bonds, and ionic interactions. And then common examples that do show up on the MCAT are the hemoglobin protein. Recall the example we gave earlier with sickle cell disease. And then you also have insulin, which is composed of 2 subunits joined together via disulfide bonds. All right, now let's talk about the denaturation of proteins. So when we talk about the denaturation of proteins, we're typically referring to the unfolding process of the tertiary and secondary structures. So we're not really referring to the peptide bonds being broken when we're talking about the denaturation of proteins. So typically that primary structure, the polypeptide, is preserved because it's a lot more difficult to break those covalent peptide bonds. And now this is achieved by exposure to urea, extremes of pH or temperature, or even some organic solvents. And in most cases, this process is irreversible, meaning once you denature a protein, it's difficult for it to assume its 3D conformation once again. So again here, just reiterating, notably the primary structure is typically preserved. Although you can hydrolyze the peptide bonds, you will need to expose them to extremes of heat. So I'm talking about like more than 100 degrees Celsius and like extremes of pH over an extended period of time, like over a 24-hour period. So now let's talk about the sequencing of proteins. And here we're specifically referring to the sequencing of that primary structure, the polypeptide. So you've unfolded your protein and now you want to determine what amino acids exist within your protein. So you can use cation exchange chromatography if you want to analyze the polypeptide in no particular order. What does this mean? So basically, you're going to hydrolyze all of the polypeptides, again, by exposing them to extremes of temperature and pH for an extended period of time. And then that's essentially going to give you a soup of free-flowing amino acids, and then you can analyze those or separate them based on charge using cation exchange chromatography, and then analyze each amino acid. And now you know exactly what amino acids are present in your protein, but unfortunately they're not in the order, that N to C order that sometimes you want to know. Now if you're looking to sequence your protein in order, so from the N to C terminal, and know exactly where each amino acid is positioned, you can use something called the Edman reagent, also known as the phenyl isothiocyanate, PITC. And I think it's clearer, rather than reading these bullet points, to just show you this schematic over here. So this is Edman's reagent, and what happens is you get your polypeptide sequence from N to C, and Edman's reagent is going to bind to your amino group, so your N terminal, and then is going to cause instability in the adjacent peptide bond right here. So now you can selectively cleave that specific peptide bond without affecting the rest of the polypeptide. So you're left with your amino acid, your very first amino acid over there, and the remainder of your polypeptide. Now you can analyze this amino acid and then repeat the process. And by this process, you know exactly the order of your polypeptide amino acids from N to C. All right, now let's move on to the misfolding of proteins. So this topic is sort of broadly discussed on the MCAT. You don't need to know the dirty details of it, but what you want to know over here is that when you have misfolding of proteins, you can get certain disease states such as Alzheimer's disease, prion disease, and Creutzfeldt-Jakob. Now, you don't need to know anything about the clinical aspects of these diseases. We'll leave that for medical school, but you do need to know that the misfolding of proteins and the aggregation of beta sheets is involved in the mechanisms of these diseases. If you found this video helpful and like the way I teach, I'd recommend watching the rest of the playlist. I spent a lot of time making sure that the videos build on one another, so it makes sense to watch them in order. Also, don't gatekeep these videos. If you have a friend who's currently studying for the MCAT, be sure to share the playlist with them. It really helps the channel grow, and I would really appreciate it. Also, did you know that every single video has customized Anki cards associated with it that cover the concepts that we discuss in a video? So if you want a way to be able to actively review the material and retain it for the long run, I've got you covered. If you're interested in accessing the Anki cards or even the slide decks within the videos, then check out our Patreon or reach out to me directly for alternative ways to access the Anki cards and the slide decks. Finally, these videos, at least as of now, are sponsored by you. So if you're interested in supporting me to be able to continue to make these high quality MCAT playlists, you can support me either via Patreon or by buying me a coffee. Links in the description down below, as well as the links for everything else, including the link to our growing Pre-Med Companion Discord community. Thanks for watching, and I'll see you in the next video.